So if you go to Canvas now, I'm finally starting to get all of the videos from the class lectures online and stuff. So you can see this thing that says list of available lectures and corresponding class notes. If you have, and I've got several up here, if you have missed lectures now, these are available and you can go watch the lectures and get credit for the participation. But in order to accomplish that, you will have to watch the video, and you can see that, click here to see the video that accompanies. If you miss this lecture, go here to the participation assignment. If you go to the participation assignment, it'll give you a little worksheet that you have to fill out as you watch the video. But if you've missed any of these lectures now, you'll start to see them appear as I assign them to you because you've missed those lectures. Okay, so if you've missed lectures in the last little while, you'll start to see assignments appear for those where you'll have to go and watch the video and turn in a little form for me that shows me that you've watched the video. Okay, so again, all you have to do is go to your Canvas page. It'll tell you there's this list of lectures. Oh, come on. And here's the list. There will be a lot more that appear up here fairly shortly. Okay, so I'm finally starting to get that up there. So we were talking about meiosis. And we had, so I guess that would require,
you got in the back of these.
So we had gotten to page 4. Again, remember, I told you that we covered this part of it life cycle in most animals that reproduce sexually. We covered that part of it in a previous discussion, so that part of it we aren't doing. That was on page 2 and part of page 3. And we had gotten down to the bottom of page 3 here to where we're going to start talking about meiosis actually. And the easiest way to remember everything that's going on regarding cell division, if it's mitosis versus meiosis, is to partly remember what the purpose of each of those types of cell division is. Okay? If I need simply to increase the number of genetically identical cells, if I need to increase the number of genetically identical cells, I undergo mitosis. And this is for development and growth, right? From the zygote, from the time that I'm a fertilized egg, one cell, I divide by mitosis. I have genetically identical cells for everything through the rest of my life, except for the one thing where I need to have a process that creates sex cells, okay? So when I need the process to create the gametes, we said that the reason that I have to go or use this special process of meiosis is that I need to go from a single or two sets of chromosomes to a single set of chromosomes. What did we say would happen if I did not do this? What would happen to the number of sets of chromosomes if we did not do this? Every time we had reproduction, the new progeny would have double the number of chromosomes it was supposed to have. First, I would have 4 sets of chromosomes, right? And then those individuals would have 4 sets of chromosomes in their sperm and egg cells, and then their offspring would have 8 sets of chromosomes, and then their sex cells would have 8 sets of chromosomes, and their offspring would have 16 sets of chromosomes, right? So I have to go through meiosis because it takes me from a diploid set of chromosomes to a haploid set of chromosomes. Okay? So a process of cell division in which a cell with a diploid set of chromosomes, 1 set from mom and 1 set from dad, gives rise to 4 haploid cells with 1 set of chromosomes, in which the set of chromosomes is a mixture of both mom's and dad's genetic material. So we can sort of compare these two processes in their features and see where they're similar and where they're different. And I'm not going to have you recreate this form like I do a lot of other graphic organizer types of things, but I am going to be able to ask you specific questions about this, okay? So we want to compare the two processes before we actually go through meiosis. We want to compare these two processes. First of all, in mitosis and meiosis, where does the duplication of the genetic material occur? It occurs in the S phase of the cell cycle, right? That's what S stands for. It stands for synthesis. So whether I am going through a cell division that gives me a genetically identical set of cells or non-genetically identical sets of cells with 1 set of chromosomes, I start from the same place. Okay, I start from the same place. And normally, whether I'm in both of these, I start from the place of having 2 sets of chromosomes of 23, 1 set from mom and 1 set from dad, going to 4 sets of 23 when I undergo synthesis, right? I normally have 2 sets of 23. I normally have 2 sets of 23. I start with diploid cells, and then I undergo synthesis, and I have 4 sets of 23. Okay? And each of the 46 chromosomes now appears as duplicated chromosomes. Okay? So we'll look at what that looks like under the microscope here in just a second. Okay, so I start from the same place for both of these. I start from the same place. But the division of the cells results only in 2 new cells for mitosis, each with 2 sets of 23 chromosomes, and they're diploid, and they're exactly genetically identical. But in meiosis, I get 4 new cells, and I only have 1 set of chromosomes, right? That's the goal of meiosis. I go from a diploid cell to a haploid cell because I have to. Okay? And the other main difference here in the division is that not only do I get 4 sets or 4 cells with just 1 set of chromosomes, the chromosomes are not genetically identical. They're a mixture of mom and dad's chromosomes. Okay? They're a mixture of mom and dad's material. So, for example, chromosome 1 in each of the 4 cells is a mixture of mom and dad's chromosome 1s. And they're not identical mixtures, right? They're unique, which is important for genetic variation, right? Number of cell divisions. In mitosis, I only undergo 1 cell division. We simply have an equal division of the duplicated genetic material. I duplicate the genetic material, and I separate it equally into 2 daughter cells. In meiosis, however, I have 2 cell divisions. I have to do this in order to get 1 chromosome or 1 set of chromosomes in each cell. In the first, we'll find out that the first division results in 2 cells, and each will have a duplicated set of 23 chromosomes, 2 sets of 23 that are a genetic mixture. And then the second division divides these 2 sets of chromosomes into single sets of chromosomes. Okay, so I need you to be aware of those features. Okay? So, I'm assuming that you have seen pictures of sets of chromosomes before. Is this true? Mm-hmm. Okay. Anybody remember what these things are called? What this picture of a set of chromosomes like this is called? Yeah.
Is it a phenotype?
It's not a phenotype. Phenotype is a genetic term for what the organism looks like. But type is correct.
Karyotype.
Karyotype. What's karyo mean? What's that? No. Where else do you hear the word karyo? When we're describing types of organisms, like pro and eu. Eukaryotic and prokaryotic, right? The word karyo means nucleus. Prokaryotic means before the nucleus. Organisms that don't have a nucleus occurred before the nucleus. Eukaryotic, the word eu means true. So true nucleus. Eukaryotic organisms have a true nucleus. So a karyotype is a picture of the nucleus. Okay, so a karyotype is an ordered representation of a complete set of an individual organism's chromosomes as seen through the light microscope using any of a number of staining techniques. Above are depicted 2 standard karyotypes of a male human. In the left karyotype, how many chromosomes do you see in this karyotype here? How many chromosomes do you see?
47.
47? Let's try it. So I see 2 in each, right? 1, 2, 3, 4, all the way up through 22. And then I see an X and a Y chromosome giving me a male phenotype, right? So this is a male phenotype because I've got an X and a Y chromosome. But I've got 22 pairs, so that's 44, 45, 46 chromosomes altogether, right? So on the left side, I've got 46 chromosomes. On the right side, what's happened to each of these chromosomes? They've now become what? I don't just have 2 chromosomes anymore. I have 2 pairs of chromosomes, right? So this is the situation after we've undergone synthesis. After we've undergone synthesis, now I have duplicated my chromosomes. Okay? And this is the point where both mitosis and meiosis start. Here is my diploid cell before I undergo synthesis. I just have 23 pairs of chromosomes. One of these is mom's and one of these is dad's, and you can't tell which. But let's just say that's dad's and that's mom's. Oops. We have the little male and female symbols there, right? So that's dad's and that's mom's, except it's not letting me draw the little arrow there. I don't want that. There we go. So let's say that's dad's and that's mom's. And so I have dad's chromosome 1 and mom's chromosome 1 and dad's chromosome 2 and mom's chromosome 2 and dad's chromosome 3 and mom's chromosome 3. I have a single set of 46 chromosomes, or mom's and dad's 23 chromosomes. And we can't tell, well, actually we can tell. The X chromosome had to come from mom, right? Because the Y chromosome had to come from dad. So this is a male. Okay, and as far as we can see, it looks pretty normal. This is a normal set of chromosomes. But once I undergo duplication, this chromosome is now duplicated to this, because I have 2 chromosomes there. This chromosome is now duplicated, and so I have a total of 4 sets of 23 chromosomes. Here I have 2 sets of 23 chromosomes. Here I have 4 sets of 23 chromosomes. And this is the point where both mitosis and meiosis start from. Okay? In mitosis, I just put these all at the metaphase plate, all 23 of these pairs of chromosomes, I put them at the metaphase plate, and they just separate, right? They're genetically identical. And all I do is just separate this one into one cell and the other one into one cell, this one into one cell, and the other one into one cell. So one of these is mom's duplicated chromosome 1, and one of these is dad's duplicated chromosome 1. Okay? So above are depicted 2 standard karyotypes of a male human. In the left karyotype, we see 46 chromosomes. How many pairs of chromosomes do you see? You see 23 pairs. Okay? Recognize that each pair comes from mom and dad. The banding pattern that you see in the chromosome is caused by the particular type of staining used. So if I use a different stain, I'll see a different pattern of banding. That's what this is called. You can see the light and the dark areas. And before we had this genetics explosion of being able to sequence the DNA over the last 20 to 30 years, this was as good as we could do. We could look at the chromosomes and we could see that maybe this dark band and this white tip here were not present on a chromosome. And that would tell us that there was a chromosomal abnormality that was present. Before all of our DNA technology was developed, that was as good as we could do. We could only see large problems with the chromosomes. We couldn't see individual genes that were mutated. But now we can. But not with this kind of a technique. Okay? So notice the banding pattern you see in each chromosome is caused by the particular type of staining, and it's utilized to help determine if portions of the chromosome are missing or duplicated. Notice the X and Y chromosome. This is the 23rd pair. These are usually called what kind of chromosomes? The X and Y chromosomes are called what kind of chromosomes? Those are the sex chromosomes. As a group, the other 22 pairs, do you remember what we call the other 22 pairs of chromosomes? You may not have heard this term before. These are called autosomes. Okay? Anything that's not a sex chromosome is an autosome. And then I asked, did you notice anything different about the karyotype on the right compared to the left? We've already explained that the karyotype on the right is duplicated. Okay? And it's not just duplicated, right? It's replicated. It's perfectly duplicated. On the karyotype on the left, standard set of 46, 23 each for mom and dad. This is the set of chromosomes that would normally be present in a blank cell. We're going to say a G1 cell. Okay, this is before synthesis. This would be the normal G1 state of any body cell other than sex cells. On the right is a karyotype of a duplicated set of chromosomes after synthesis. Notice there are 92 total chromosomes, a duplicated set of 23 from mom and a duplicated set of 23 from dad. Again, this would be the state of meiosis, or the nucleus at the beginning of either mitosis or meiosis. And now we need to remember the terminology. Each of the chromosomes in a duplicated pair is called a sister chromatid. Although remember, it is a chromosome. So in the non-duplicated state, this might be mom's chromosome 1, and this might be dad's chromosome 1. In the duplicated state, this might be mom's chromosome 1 and dad's chromosome 1. But now each one of these things is called a sister chromatid, right? Okay, so each one of these things is a sister chromatid, and this thing on the left is a chromosome, and this thing on the right is a chromosome. And we've called this point of constriction at the center, we've said this several times already, what is that?
Centromere.
That's the centromere. Okay, and we said in mitosis that these things are all lined up at the metaphase plate. Okay, this might be mom's chromosome 14, and this might be dad's chromosome 7, and this might be mom's chromosome 6, and this might be mom's chromosome 19, and this might be dad's chromosome 1. Okay? And all of these are lined up, right? There are 46 pairs of these all lined up, and we said that at metaphase, when everything's all set and ready to go, we've got microtubules hooked up to each side of this. And remember that each chromosome, before it's exactly where it's supposed to be, it's sending out signals. It's saying, I'm not ready to divide yet. I'm not ready to divide yet. I'm not ready to divide yet. But at that moment that they're all lined up exactly on the metaphase plate like they're supposed to be, then all of these things stop sending the signal, I'm not ready to divide. And at that moment, each one of these things splits in half. And one goes one way and one goes the other way. And that's how we get exact numbers of chromosomes in each cell, right? We said that if we didn't have that mechanism where each one of these wasn't constantly sending out signals that, hey, I'm not ready yet, cell division could start before we were ready, and maybe one of these that wasn't ready yet, one of these that didn't have an attachment on one side, the whole thing would go into one daughter cell, and that would be a problem. Okay? So think about it. If I just have the fertilized egg here, and it's ready to undergo that first cell division to get two cells, right? We're going to get two daughter cells. But this happened where maybe both of the chromosome 14s from mom went into one of these cells, and none of mom's chromosome 14 went into this cell. Half of my cells would now have 3 chromosome 14s, and half of my cells are from mom, and half of my cells would now have no chromosome 14s from mom. Or I'm sorry, 2 chromosome 14s and no chromosome 14s. Half of my cells would be without a chromosome 14 from mom. They'd still have a chromosome 14 from dad, but they wouldn't have a chromosome 14 from mom. Okay? And that many of my embryo cells, 50% of my embryo cells without a chromosome 14 from mom would be lethal. Okay? So there are lots and lots of things that can go wrong with this process, that even after fertilization occurs, the embryo can end up in a lethal state. It's estimated that probably as many as 60 or even 70% of all conceptions don't make it. It's just that we don't recognize it because they don't make it at such an early stage that the mom doesn't recognize that she's pregnant. Okay? So probably over half of all conceptions never make it to the point where mom recognizes that she's pregnant, because this process is so complicated. Okay, now here's a new term. Duplicated pairs of chromosomes of the same chromosome number are called homologous. 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 What I mean by that is if I have mom's duplicated chromosome 14 and I have dad's duplicated chromosome 14, these are homologous. So duplicated chromosome 3 from mom and duplicated chromosome 3 from dad are homologous. Duplicated chromosome 17 from mom and duplicated chromosome 17 from dad are homologous. So I could go back up to this set of chromosomes and say, these are homologous. These are homologous. Okay? But if I had mom's 4 come on. And dad's 9. These are not homologous. Okay? And this is going to be an important term for us. These are homologous chromosomes, and these are not homologous chromosomes. Okay? And here's why this is important to us. This is why this is going to be important to us. Because in mitosis, let's say that this is mom's duplicated chromosome 3, and then let's say this is dad's duplicated chromosome 17, but then I've got dad's duplicated chromosome 3 here. Okay? In mitosis, I want these to be lined up like this, each duplicated pair on the metaphase plate. I want all of them to be lined up singly like this. I want mom's duplicated 3 and I want dad's duplicated 3 because I want one chromosome to go each way. I want genetically identical chromosomes to go into each daughter cell so that this daughter cell gets one of mom's chromosome 3s, and this cell gets one of mom chromosome 3s and dad's chromosomes 3. But in meiosis, the goal is different. In meiosis, instead of metaphase being this, the first metaphase is going to be this. This is going to be mom's 3, and this is going to be dad's 3, and this is going to be mom's 17, and this is going to be dad's 17. Our first metaphase in meiosis, we don't want this. We want homologous chromosomes to be on the metaphase plate. We want the homologous pairs to divide first. Okay? So I want a pair of chromosomes to go one way and the other pair to go the other way. We're going to have to plug in here.
So, do the homologous chromosomes come from different parents, like dad chromosome 17 and mom?
Good question.
17?
Where is my... I brought it in with me. Where did I put it?
was your question again, please?
For a chromosome to be homologous, does the pair have to come from the same parent, or?
No. No. Right? Because one of these is mom's and one of these is dad's. So these are homologous chromosomes. But instead of having individual chromosomes line up on the metaphase plate, in meiosis, the first thing we want to have is the homologous pairs to line up on metaphase plate. We're not going to divide these individual chromosomes yet. We're just going to divide the pairs. Okay. We're just going to divide the pairs. But what do you remember has happened in prophase to this homologous pair of chromosomes? You remember what happens in prophase 1 of meiosis to this pair of chromosomes? Why is it still even attached together? How is it going to line up together with its partner from dad? What happened to it in prophase? You remember? Anybody remember what happens in prophase 1 of meiosis? What happened to these two pairs of chromosomes in meiosis 1, or in prophase 1? Do you remember? Huh?
It wasn't them splitting, was it?
No. If this is chromosome 3 of mom, and this is chromosome 3 of dad, they got together in prophase. They attached to each other and switched genetic material. Right? In prophase 1, the reason they can line up like this instead of like this is because they're stuck together because they switched genetic material in prophase 1. And they're not unstuck yet. They're not unstuck yet. They're still stuck together where they switched genetic material. Okay? So, the whole reason we want to have the term homologous is because it describes what happens in the first meiotic division. Homologous pairs of chromosomes line up on the metaphase plate in meiosis 1, whereas in metaphase 1, whereas in simple mitosis, all of the individual duplicated chromosomes line up. The pairs don't line up. All of the duplicated chromosomes line up, and they're all going to go one way. Okay? So let's keep this term homologous in our mind here as we try to make more sense out of this. Okay, so we've got this new term, homologous. Duplicated pairs of chromosomes of different chromosome numbers are called non-homologous. Okay? Duplicated 4 from mom and duplicated chromosome 11 from dad are non-homologous chromosomes. And I don't really need you to go back and circle those. We already did something like that already. Okay, so now we need to talk about the first meiotic division, not the first mitotic division. The first division of meiosis is meiosis 1, and it begins from the same place that the mitosis begins, right? We have 46 duplicated chromosomes, each consisting of a pair of sister chromatids, 92 chromosomes altogether. So, although the phases of meiosis have many similarities to the phases of mitosis, the goals of meiosis are different, right? With mitosis, my only goal is to create genetically identical cells. That's my only goal. And the reason to do that is just simply to increase my cell number, either because I'm growing and developing or because I'm healing. But in meiosis, I have to reduce the chromosome number from diploid to haploid, and I have to introduce genetic variation. So in prophase 1, we still have the following goals when we're looking at it microscopically. I've got to break down the nucleus. Okay? All of my chromosomes are attached to the nuclear membrane. I have to get rid of the nuclear membrane. I also need to get rid of my nucleolus. I can't make proteins during meiosis because my chromosomes are condensed. So there's no reason to have the ribosome factory, right? The ribosome factory is the nucleolus. So I get rid of the nucleolus, I get rid of the nuclear membrane because I have to. Okay? And I condense the chromosomes, right? I turn my DNA in from two balls of fishing wire that are meshed together and I can't separate. I turn those into chunked chromosomes that I can easily separate. But in prophase, they're randomly placed, right? They're not lined up yet. And my spindle apparatus is forming, just like in mitosis. But ultrastructurally, things are a little bit different. Instead of just simply condensing and hanging out until I'm ready to go at mitosis, each duplicated pair of homologous chromosomes forms a physical association. Okay? They form a physical association. I have two pairs of chromosomes that are homologous. One, again, might be mom's chromosome 3, and one might be dad's chromosome 3. Okay? And they swap genetic material. Okay? They overlap with each other, and they exchange genetic material. This is an ultrastructural picture of that. I can see that both of these chromosomes here are overlapped. And I might swap this segment of DNA for this segment of DNA. Okay? I might trade those two places of pieces of DNA. And I might have another piece of DNA here that I exchange for a piece of DNA here. Okay? Imagine trying to do this and it has to be exactly the same piece of genetic material. Okay? It has to be exactly the same piece of genetic material. If I'm off by one base pair here, I screw everything up. Can you get your leg off my table, Embry? This is not a lounge. Okay? I screw everything up if I'm off by even one base pair. Okay? How difficult is it for us to even be alive? Or to have been created or born, right? It's extremely difficult because this process has to be exact. It can't be off at all. So each of these paired homologous chromosomes swap genetic material. Okay? And because this is composed of 4 chromosomes, it's called a tetrad. Not tetrus, but tetrad. In this physical association, paired homologous chromosomes line up next to each other and exchange genetic material. Points of genetic exchange are called chiasmata.
Oh.
Okay, where I am exchanging genetic material. Chiasmata is the plural term, chiasma is the singular term. So this point of exchange right here is called a chiasma.
Which is also a form of poetry.
Is it?
Yeah.
Cool. I was not aware of that. Okay? And where I've exchanged genetic material, that's called a recombinant. It's recombined. Only one chiasma is illustrated here, but many occur per pair. Okay? So these things happen in prophase. Okay? In prophase of mitosis, they're just waiting, they're hanging out until a microtubule attaches to them and pulls them to the metaphase plate, right? Here, they're still waiting for that too, but they're also exchanging genetic material at the same time. This is how we shuffle the deck. This is how the genetic material gets exchanged between mom and dad's chromosome. So when these things line up at the metaphase plate, the homologous pair, I don't really have a mom or a dad's pair of chromosomes anymore, right? Both of these pairs of chromosomes now contain a mixture of genetic material, and it's a unique mixture of genetic material. It's not pre-programmed in any way, shape, or form. It's entirely unique. A unique mixture of mom's and dad's genetic material. So, this exchange of genetic material mixes the genetic material of that individual's parents, resulting in an extraordinary number of possible new combinations of genetic material to be subsequently placed in sperm cells or ova. So, in meiosis, another goal of prophase 1, instead of just breaking down the nucleus, condensing the chromosomes, and forming the spindle, we also have to shuffle the genetic material. We have to recombine it. So now, not only do we see the nuclear membrane broken down, chromosomes condensed and randomly placed, no nucleolus, we also see formation of chiasmata with exchange of genetic material. But the spindle apparatus is still forming just the way it was before. Okay? So, following this process, recognize that none of the 4 chromosomes originally present in each duplicated pair is now in its original form. All 4 contain some genetic mixture of the individual parent's DNA, and each of these 4 chromosomes will now be separated into 4 sex cells, sperm in the male, ova in the female. Okay, and you've got this diagram. Right here we have this association. What would happen if the wrong homologous chromosomes got together? What would happen if what would happen if this homologous chromosome right here was actually, let's say, mom's 4, and this was dad's 16? That would be disastrous, wouldn't it? Okay? That would be disastrous. We're not very sure how these pairs of homologous chromosomes find each other. But in some way, shape, or form, mom's chromosome 4 and dad's chromosome 4 have to find each other in order for this to work. Okay? There's still a lot of research going on to help figure that out. And so it's not the individual chromosomes that line up during meiosis in metaphase 1. Remember, in mitosis, we would just have all of the pairs line up. Okay? We would just have all of the pairs line up. But we would have all of the duplicated chromosomes line up like this. and one would go one way, and one would go the other way, and one would go one way, and one would go the other way. But in meiosis, we've got pairs of homologous chromosomes that line up because they're still stuck together. Okay? That's how we make this work. They're still stuck together at this time, so there's no chance of a mistake that I can get one of these going one way and the other one also going the other. Well, we can get that happening. There could be a mistake where these don't separate and all four go into one daughter cell. But, man, there's so many places where there could be mistakes. And then the homologous pairs get separated into a daughter cell. Okay, so you can see that the homologous chromosomes separate, and this is anaphase 1. Okay, so a lot of similarities, but major differences. In prophase 1 in mitosis, there's no crossover. There's no association of homologous pairs of chromosomes. In prophase 1 of meiosis, there is a physical association of homologous chromosomes, and recombination occurs during that time. In metaphase 1, I have lining up of homologous pairs in meiosis. In metaphase of mitosis, I just have lining up of the individual duplicated chromosomes. In anaphase 1, I separate the homologous pairs. So, real quickly again, in metaphase 1, the exchange of genetic material during prophase 1 accomplishes the goal of shuffling the genetic material, and is one of the major differences between prophase 1 of meiosis and prophase of mitosis. A second major difference occurs in metaphase 1. Okay? Again, I need to attach spindle fibers to the pairs of chromosomes and move them to the metaphase plate. The chromosomes line up at the metaphase plate. The spindle feature is completed. In mitosis, all duplicated chromosomes line up on the metaphase plate, each of 23 moms and each of 23 dads, so that each daughter cell can receive all 46 of the identically duplicated chromosomes. Each daughter cell will then be genetically, or will be diploid and genetically identical. In contrast, in metaphase 1 of meiosis, each of the duplicated homologous chromosomes remain paired, and the pairs are brought to the metaphase plate and lined up. And you can see that what will get separated into daughter cells is not identical individual sister chromatids, but genetically mixed pairs still attached at the centromere. Each daughter cell will now get 46 chromosomes. They will be diploid, just as in mitosis, but in this case, it will be 23 pairs of attached mixed chromosomes rather than 46 identical chromosomes. So the goals of metaphase 1, pairs of homologous chromosomes are attached to spindle fibers and moved to the metaphase plate. And I see homologous pairs line up at each metaphase plate. I know we're getting close on time here. Now, yours didn't copy correctly. For some reason, they're weird. But this is what they should look like. Here in mitosis, chromosome 3, chromosome 16, chromosome 16, chromosome 3. One is mom's and one is dad's. And just the individual things go one way or the other way. Here, I have the pairs that are lined up, and the homologous pairs are separated. Okay, so in meiosis 1, we separate pairs of homologous chromosomes. In mitosis, we separate just duplicated chromosomes. Okay? The other thing about this is, and this contributes to the genetic variability here. Let's say that this was originally mom's chromosome 3 and this was originally dad's chromosome 3. Obviously, they're mixed now, right? But let's say this was originally mom's 3 and this was originally dad's 3. But I don't have to have it that way. I could have had mom's chromosome be over here and dad's chromosome be over here. And this is called random assortment. Random assortment. When I line the homologous pairs up on the metaphase plate, whether this was originally mom's or originally dad's or originally dad's and originally mom's, is random. And this adds to the genetic variability. And it's specifically called random assortment of the homologous chromosomes. Okay? So that's an important term, random assortment. So not only do I separate homologous chromosomes, it's a random separation. Okay? I have some questions here that we need to answer so that we have full understanding, but it's going to take a little bit longer to discuss these questions than what we have left, so we're going to stop there. Is this making any sense at all? Or is it just entirely confusing? Did you understand meiosis to this degree when you heard it the first time? You don't remember meiosis from biology class? I'm sure Sperry taught you about it.
That was like the freshman year.
So?
A while ago. Do you know how many other classes I've had to absorb information from? Meiosis was
Okay, I learned about meiosis decades ago, and I still remember it.
You teach it every day.
But as a student, it's your job to know it too.
To pass my classes and to be able to know enough about each class.